So today I'm going to be talking to you about the threat that parasites pose to wild bees. So just some background onto the importance of pollinators. Insects are responsible for about 2/3 of plant pollination. And when we look at this economically, it's worth about $153 billion each year just for pollination services. So they're pretty vital in our high-value crops. And so I'm sure you heard about colony collapse and the whole Save the Bees thing. And pollinators have been shrinking in size and in range. It's not just honeybees, it's other insects as well. And part of this is thought to be due to generalist parasites that can infect multiple hosts, and that's what we're going to be talking about today. So just to highlight the importance of these pollinators, the wild bees that we're looking at, most of them are solitary bees, and highlighted here in this column are these solitary bees. And you can see that when we're looking at pollen loads and how effective they are at collecting, in comparison with the honeybee apis, they're as effective, if not more effective. So they're pretty crucial in our pollination. Just to start, here's our sweet little solitary bee coming out of its home. So some things that we know about bees, there's about 20,000 species globally. And then just within New York State alone, we have 416, with 412 of those being wild bee species. On the next slide, I'm going to show you a pollinator network, but just to these 2 points here, looking at wild bees and at wildflowers, we're seeing traces of honeybee parasite DNA. And so we're interested to see whether or not these wild bees are becoming infected. And so we're looking at these flowers as well because they're acting as parasite transmission hubs, where the parasites are actually staying on the flowers. So what we don't yet know is whether or not these honeybee parasites are being transmitted to the wild bees. And so with that comes a whole slew of other questions. So if they are being transmitted, what species are becoming infected? And with infection, is there any impact on survival? And then if it's not being infected, are they perhaps a non-host vector and able to transmit this parasite further? So here's this really great network that our lab did, looking at, on the left-hand side here are the wild bee species, and on the right are the wildflower species. And all of these circles are showing a positive result for honeybee parasite DNA. And the reason that we're looking at a honeybee is because we can see here all of these things that they're visiting. And we know quite a bit about a honeybee because they're managed bees. So people are really interested in those, and they're easier to study because we have them. So yeah, this is really just to make a point of how complex the pollination network is and the amount of crossover that could happen with any of these wild bee species to any of these flowers that this honeybee is foraging on. So really quickly, this honeybee parasite that we're looking at is known as Nasima. It's here on the left. There are these spores. It's a microsporidium parasite that lives within the gut. And here's a honeybee gut. And so we know that Nasima is transmitted fecal-orally. So a honeybee's foraging on a flower, maybe it defecates, leaves. Another bee comes along and as it's foraging, perhaps ingests some of those spores. And we want to know if this is causing an infection or having any impact on these wild bees. But we do know that some of the symptoms of Nasima, at least within honeybees, causes dysentery and shortened survival. So we want to know if that's happening elsewhere as well. This is an overview of the diversity that we found. So we sampled from 3 different sites around this central New York region. We found 14 genera, and here's just your friendly neighborhood carpenter bee. Here's a bombus, a common eastern bumblebee, and then here highlighted is Ceratina. This is a sweat bee that's probably in your backyard. And we found quite a lot of these, so I just want to highlight, we'll be talking about this later as well. As far as our experimental method went, we were not discriminate in the bees that we were sampling. Anything that we found, we collected. We did not collect honeybees, as we were not interested in those. It was just the wild bees that we were finding. We ended up getting 161 for the Nasima treatment. And upon collection, we would separate them by genus and between treatments of sex so that we would have an equal separation. So here we use these feeders to serve as a way to administer the inoculant. And in here we have a 40% sucrose solution in combination with a bee gut homogenate. So for the control treatment, we were giving them bee gut homogenate from uninfected honeybees, and for the Nasima treatment, we were using bee gut homogenate of infected honeybees. And again, mixed with this sucrose so that they would ingest it. And so here I have highlighted this little blue dot is a defecation event of a bee. And so we were really closely monitoring defecation of the bees this summer to know whether or not they were ingesting the inoculant since we weren't administering it directly. And we were going to screen these feces at a later date, we didn't get to that yet, to see if there are spores present. And here's just a comparison in the variation of feces. I became very familiar with bee feces this summer. So here's just a timeline of how this experiment was done. Here we are collecting the bees. 3 days after collection, we're going to inoculate them. And this is a really important day for us to look at the feces to know if these bees are coming in with any sort of outside infection. And so we can see what's happening in nature. And then here, just most of this experiment is basically maintenance. So every 3 days, we're collecting feces. We're refreshing their containers and giving them food. But these feces samples are going to be really important too. If it's increasing, then we're seeing that maybe the spores are reproducing within the bee. And then this is going to be a sign of infection. If it decreases, perhaps it's just passing through. So again, this is just some future research here. And 15 days post-ingestion, actually quite a few of our bees lived a really long time, and we had to terminate them 15 days post-ingestion. And this is another important step where we're going to dissect the bees, and then we're going to look at their guts to screen for Nasima spores. And all the data I'm going to be talking about is from the bee gut. So here, this is just basically to show the presence of Nasima that we're finding within the gut. So with all of these genera, here you can see Nasima in orange. There's a more frequency in the prevalence of Nasima treatment than there is in control treatment. And with serotina, again, that little sweat bee, we're seeing both control and treatment did have presence of Nasima. We don't know yet if that's what they came in with, if that's what they got here. We're going to figure that out with the feces. But again, this is just to show you that maybe around 40% at the highest is the presence of Nasima within these bee genera. Again, this is no significant difference simply because our sample size is quite low, but they will be expanding upon that. So looking at those genera that did show presence of Nasima, if we look at the intensity of the spore load within those genera, you can see that the intensity is higher within the Nasima treatment if it's present than it is in the control treatment if it's present. No significant difference here either, again, because of the sample size, but this is just a nice visualization to see that the spore count is quite different between the two at this point in our data. And then this is just a fun little plot. Again, our serotina, you can see that the mean intensity here for Nasima, around 30,000 spores. Again, this is just a treatment size of 10, so no significant difference. But this is interesting, and hopefully this will flesh out with a larger sample size. And then just to make a point on this, honeybees, when they start to show signs of an Nasima infection, it's more than 1 million spores per bee. But we don't know what a typical spore count would be for a serotina. That's a smaller bee. So that's going to be interesting to see. Maybe 30,000 is a full-blown infection for serotina. Hopefully we'll figure something out about that as well. And then just to point out survival here, again, no significant difference between the treatments. But just to show you, most of these bees were living the full 15 days post-ingestion, like about 90% in some of these. Andrina a little bit lower. But something really interesting here with Halictus, maybe we might see this with a larger sample size as well. There was a presence of Nasima in both control and treatment, yet it didn't impact survival. This is maybe a sample size of 7 per treatment, but that's quite interesting. And then just sort of to wrap up the importance of this study, we don't know anything about wild bees in reference to how parasites could be infecting them. So just knowing whether or not this could have a serious impact is really necessary to understanding conservation. So with these wild bees, it's not necessarily like honeybees. If a hive dies, they're managed, you can sort of farm those. But with a wild bee, if that decreases, this becomes a whole conservation effort. So it's just really important to better understand what's happening with them pollinator health-wise. And so this project that I was part of this summer is part of this larger project, the EEID project, looking at infectious diseases, pollinators. And so after I leave, they're going to be looking at other managed bee parasites. They're going to be looking at parasites found in bumblebees and how those are impacting wild bees. And just increasing the sample size, they're looking at flight cages. There's a whole bunch of stuff happening within the McArt lab and around institutions across the country to better understand our pollinators. So then just to reiterate why this is important in a plant science setting, without these pollinators, we would really be struggling with agriculture. And with food security being such an issue right now, too, with our increasing population. We really need to put in some time and effort to understanding these sweet little guys. So if you want to help them, you can plant a wildflower garden. Just sort of keep your eye out for them. So here's just a huge thank you to my lab. It was absolutely wonderful this summer. I had such a great time. And then just a thank you to Boyce Thompson for the REU, NSF and USDA for funding, and Cornell for the opportunity. Any questions?
I have a question. So
Yeah.
you talk about honeybees infecting the wild bees. Is it also possible that the wild bees are a reservoir that I keep getting my hives infected? You know, that if the wild bees are resistant, that they are infecting the honeybees. Is that a possibility?
I mean, I don't know, simply because we don't have a lot of information on wild bees, but since honeybees are so commonly looked at, we know that they are predominantly found there. And since we know so much about honeybees in reference to the wild bees, perhaps they could be. It's hard to say. It's hard to say for me. Maybe Scott would know. Yes.
Do you think there's other pollinators that could be affected by these pathogens?
Absolutely. So there's actually another project happening in our lab with hoverflies. And as you see, let me show you this again. In our effective pollinators. Here, these are all flies. And so they're looking to see how hoverflies could potentially be impacted by parasites of managed bees as well. And they're getting some pretty cool results as well. Yeah.
So we should probably move on in terms
Cool.
of time.
Thank you.